AMALearning, you're about to. Specification, point 1.3. Explain how changes in microscope technology, including electron microscopy, have enabled us to see cell structures and organelles with more clarity and detail than in the past, and increased our understanding of the role of subcellular structures. To successfully answer any GCSE question linked to this specification point, you will need to be able to compare the light and electron microscope. The most common question requires for you to state that the electron microscope has a greater resolution and magnification. Why has our knowledge of the role of subcellular structures improved? There have been developments in our microscope technology. Electron microscopes have enabled us to see cell structures and organelles with more clarity and detail than in the past. It cannot be used to view live specimens. What are the differences between electron microscopes and light microscopes? Electron microscopes are larger and so not portable, more expensive, difficult to use and require monitors to view the image, unable to view live specimens. Light microscopes are smaller and portable, more affordable, in fact they are relatively cheap, much easier to use, able to view live specimens. An electron microscope has a greater magnification and a greater resolution. The electron microscope has increased our understanding of subcellular structures. Greater magnification and resolution is key here. It is commonly asked why electron microscopes are more advantageous or better for viewing cells. The answer: it has greater magnification and resolution, which leads to a more detailed image. To reiterate, you will get the first mark for stating greater magnification. The second mark will be given for writing greater resolution. And the third mark for adding leads to a more detailed image. It is important that you understand key terms such as resolution and magnification. And alongside this, you must know how to calculate magnification. Resolution is the ability to distinguish between two separate points. A magnification is the ability of a microscope to produce an image of an object at a scale larger than its actual size. Magnification is equal to the size of image divided by size of real object. An easy way to remember the formula is IM. If we make the size of image the subject of the equation, then the size of image is equal to size of real object multiplied by the magnification. Size of image can be shortened to I. Size of real object can be shortened to A. Magnification can be shortened to M. This simplifies the overall equation to IM, or I equals AM. Let's apply I equals AM to an exam-style question. For ease, let's initially convert this to a magic triangle. So in this case, the I goes at the top and the A and M go at the bottom. Step 1. Let's identify what information we have. 0.04 millimeters represent the actual size. So this means we know A, and 500 represents the magnification. So this means we know M. Second step is to substitute the numbers into the equation. In this case, I equals 0.04 multiplied by 500, which equals 20 millimeters. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.